Thank you very much. And at the outset, I will appreciate Dr. Banshi and the team at Ahmedabad and all the faculty of RSSDI that amidst such a problematic scenario, they've organized this conference on a short notice, although in a hybrid model. And I've been invited to speak on ominous octet and mitochondrial modulators, somewhat unusual or maybe a novel kind of topic. And at the very outset, I will also like to invite everybody to Jaipur for the upcoming EPICON, that is the Physicians Annual Conference 22, which will be held from 10th to 13th of March. And being organizing secretary, I look forward to host and welcome all the delegates who are probably listening to me. Thank you very much. So moving on to the presentation. So these are my favorite slides, although everybody knows it, but because whenever we discuss diabetes, let's know that what is the burden of the diabetes in the society, and it makes the presentation impactful, that what for we are gathering and what we are talking about. So large numbers globally, and India ranking second after China. It's a pathetic situation, but a fact. And then you can see that what numbers are growing by 2030, that there will be 115 million diabetics alone in India. And globally, if you see that, then there's almost 578 million people. So we need to know the gravity of the disease, and that's why we need to understand the pathophysiology and the new novel therapeutic approaches. And this is how the pathophysiology has evolved over the period of time. You can see from the triumvirate to omnious octet. So the same man, D. Fronzo, in 1998 gave the hypothesis of triumvirate hypothesis, where impaired insulin secretion, resistance at the level of the skeletal muscle, and the involvement of liver in form of hepatic glucose output was postulated. And later on, after maybe 10-11 years, in 2009, the same man came with another hypothesis which was accepted globally, that it converted into ominous octet, where all these organs were playing a pathogenetic role, whether it was the intestine, incretin effect, the adipocytes, the kidney, the muscle, the brain, the liver, and the islet cell obviously is in the center, because beta and alpha cells. But here, not only beta cells but alpha cells were also implicated in pathogenesis of diabetes. So combine all these factors led to hyperglycemia, and the only thing which comes to our mind is looking to this ominous octet. We have been looking this slide for last almost 10 years or more, that are we treating the tip of iceberg with glucocentric approach only. We should not be concerned only with the lowering of glucose or controlling the parameters like HbA1c, but the key problem, that is the pathogenesis, has to be addressed, and for that we need to understand the pathogenesis in detail. And why it is important, because you can see here that if you see the Indian data, then Indian diabetic phenotype, there is early progression with multiple comorbidities, and there is high prevalence of micro and macrovascular complications in young type 2 diabetes. As you can see here, that the blue and the red bar, that how early, maybe in the second decade or third decade, the various macrovascular complications like neuropathy, retinopathy, nephropathy, they set in. And not only this, they are coupled with increased burden of CV risk factors at early age, because these days we see the increased incidence of smoking, alcoholism, obesity, hypertension, and dyslipidemia, and then the stress, which the mental stress and lack of physical exercise. So all these things, they lead to complications at early age, both in terms of micro and macrovascular complications for Indian population, or patients rather. So type 2 diabetes remains progressive despite current therapeutic advances, and we need to know the reasons: adiposity and role of adipocytokines, oxidative stress and damage, higher levels of glucagon, genetic factors, and in the red you can see what I am talking about today is mitochondrial dysfunction. So probably from the organs we are moving to organelles, and we are deep diving into the pathology and going into at the cellular level rather than the organ level. And moving on to the next part of my presentation is that how the ominous octet and complications and the mitochondria is playing the role. So mitochondrial dysfunction at the level of beta cell lead to decreased insulin secretion, and it leads to increased insulin resistance. We will see later on in the slides how it is leading. So at the fat cell there is accelerated lipolysis, then there is decreased incretin levels, and then there is increased glucagon levels, and ultimately hyperglycemia, endothelial dysfunction, and vascular complications sets in. And this is the powerhouse of every cell, and it generates the energy. It keeps the functions of each cell and tissue going in our body, and if this gets damaged or altered, then the problem is anticipated. And dysregulated energy substrate metabolism in mitochondria leads to the problem, and healthy mitochondria knows how to alternate fuel switching involving glucose substrates or pyruvate or maybe fatty acids, but then this ability of mitochondria is lost, and you see that switching of metabolic fuels is lost during chronic feed, obesity, and type 2 diabetes. So this is the observation which has been seen in the mitochondria, and if you see the mitochondrial dysfunction across organ systems in type 2 diabetes, then you see that at different levels, whether it is the heart, whether it is the muscle, whether it is the pancreas, or whether it is the adipocytes, you will see that there is decreased ATP synthase, decreased mitochondrial density, decrease or maybe altered electronic transport chain expression and activity, and an increase in gluco and lipotoxicity sets in, and there is decreased nitrous oxide levels. So the endothelial dysfunction sets in. So whatever is the level, if you are seeing the organ, the dysfunction is actually at the organelle level, and it's at the micro level, and we need to deep dive into understanding of this pathogenesis at each organ, and mitochondria being the powerhouse is sitting at the center of each organ creating this form. So there are many pathophysiological pathways such as oxidative stress, inflammatory responses, adipokines, obesity-induced insulin resistance, improper insulin signaling, and beta cell apoptosis, which is associated with development of type 2 diabetes. And there is increasing evidence of the role of mitochondrial dysfunction in the onset of type 2 diabetes, and not only this, but diabetic complications also. And the role of mitochondrial dysfunction in type 2 diabetes is reviewed together with its modulation by anti-diabetic therapeutic agents, an effect that may be independent of their hypoglycemic effect. So we are not concerned about only the glucose lowering or correction of HbA1c, but correction of the pathogenesis, and maybe going or reaching up to the mitochondrial dysfunction, which probably is one of the novel targets for therapy in diabetes, can help us in a better way rather than controlling the blood glucose levels. And how the mitochondrial dysfunction is setting in: point mutations in the mitochondrial DNA, activation of PKCs. I will not go into the detail, but the mitochondrial dysfunction can happen because of these factors. So accumulation of fatty acids, diacylglycerol, long chain fatty acids like acyl-CoA activates protein kinase C isoform, leading to increased phosphorylation of insulin receptor substrate, and ultimately what it leads to is impaired insulin signaling and development of insulin resistance. Mitochondria induce oxidative stress, another problem where the insulin resistance has been linked because of this mitochondrial dysfunction, because of excessive free radical generation in cases of mitochondrial malfunction when there is impaired insulin signaling. Pancreatic beta cell dysfunction. It is postulated, we all know that 80% of beta cell mass is lost by the time the patients are diagnosed with type 2 diabetes, and mitochondria being the powerhouse which is responsible basically for ATP generation or energy synthesis of beta cells, they are having problems, and because of the generation of free radicals. Why this happens, we don't know, but probably genetic environment and environmental factors, they are playing a role, but insulin secretion is decreased from the beta cells just because the mitochondria are not working. And these are all basically not only postulations, but they have been proved, and these papers have come in the Journal of Cellular Physiology recently, maybe in 2019. So there is a strong relationship of mitochondrial dysfunction to diabetes and its complications. We know what are the causes: point mutations, which lead to upstream events where the signaling and the protein expressions are defective, and there are different anti-diabetic agents which can have a protective effect against mitochondrial damage to mitochondrial dependent oxidative change, stimulating mitochondrial biogenesis and increasing the mitochondrial mass, potentiating the mitochondrial antioxidant defense system and adjusting the mitochondrial membrane potential. And conversely, degrees of mitochondrial toxicity and mitochondrial suppression by anti-diabetic agents. So we should see or weigh that which agents are beneficial and which are not, which are only controlling the glucose, but on the other hand they are giving damage to the mitochondria at the cellular level. So mitochondrial dysfunction accounts for both skeletal muscle insulin resistance and impaired pancreatic cell insulin secretion. And you can see that I will not again go into the detail, this is a busy slide, but the crux is that increased glucotoxicity and mitochondrial dysfunction is related because of increased fatty acids which the mitochondria is exposed to in the diabetes because of the lipolysis at the level of the adipocytes, and there is decreased glucose utilization and there is increased beta oxidation at the level of, that is the crux of this slide. And again, this is a sort of a repetition, but here you can see that the mitochondrial dysfunction has been correlated with the endothelial dysfunction, and ultimately decreased phosphorylation of IRS1 and nitric oxide production is there because the mitochondrial dysfunction is there. And when endothelial dysfunction is there, you obviously we will expect that macrovascular complications or maybe even microvascular complications will arise in type 2 diabetes. And why this happens? Genetic factors, we have seen in the previous slides also, where the genetic variants, the mutations, then there are environmental factors like obesity, malnutrition, the pollutants, the endocrine disruptors, and various other drugs, they lead to mitochondrial dysfunction. So on one hand, they are causing beta cell dysfunction, on the other hand, they are producing insulin resistance and development of type 2 diabetes. So coming to the last part of my talk, that what can be the interventions or what are the mitochondrial modulators which we can target. Look this slide, this is a very beautiful slide, that in the center, the respiratory capacity, which is the mitochondria, because they allow the cell to breathe basically. So pharmacological agents, exercise and healthy diet, they improve metabolic health. And on the other hand, you can see here that obesity, aging and sedentary lifestyle, they decrease the respiratory capacity, so diabetes develops. So mitochondrial modulators are the drugs that benefit patient by novel mechanism that would make the system, tissue or organ function efficiently without altering the hemodynamics. And you can see here that mitochondrial modulation can lead to correction of metabolic dysfunction arising due to inappropriate or underutilization of carbohydrate, protein and lipid substrates. And not only this, these drugs can directly correct the metabolic defects at the level of liver, pancreas, or skeletal muscle to improve insulin resistance, and insulin sensitivity can be increased. And directly they improve cellular energetics by glucose and lipid disposal while improving nitrous oxide mediated endothelial dysfunction, thereby reducing the CV risks. So if you target the mitochondria, you can have multiple benefits beyond glucose control. So some of the agents or interventions which can improve the mitochondrial function, exercise is one. Exercise, it has been long known that exercise improves glycemic control and insulin sensitivity in obesity, thus slowing down the progression of diabetes. And it exerts its effect mainly by acting on mitochondria, where the muscle tissue adaptation to exercise includes increased mitochondrial biogenesis and mitophagy. And defective mitochondria is lost, whereas the new mitochondria or the existing mitochondria, they work efficiently. And these are the mechanisms. I will not go into the details, where the calcium or the AMP ATP ratio or the NAD/NADH ratio and the reactive oxygen species production and all these kind of things, they are altered in a beneficial way by exercise. But biogenesis, where the PGC1 alpha and AMPK activation happens because of exercise. So that is the thing which we desire, and there is a transient low intensity ROS production by physical activity, which signals, I'll just take 2 minutes more, right? So that serves as signal that promotes beneficial effects such as mitochondrial biogenesis and function, and this phenomena has been called as mitohormesis. Mitohormesis is the term where the mitochondrial function and its biogenesis can be improved. So this is the representation how exercise is helping us in improving the metabolic function. Then activation of AMPK pathway can be achieved by metformin. SGLT2 inhibitors have also shown. Then fewer newer drugs like AICAR, then experimental drugs like O304, they have all shown that AMP activation can be done and mitochondrial function can be improved. Now there are certain existing pharmacological therapies which have effect on mitochondrial function like metformin and sulfonylureas. They have been shown to be beneficial. Pioglitazone again has been shown to be beneficial, but partially because it has beneficial effects on the lipid metabolism but not on the glucose utilization. Then gliflozins, yes, they are beneficial. I will not go into the details. Insulin again is beneficial. Then SEP-MOX, which is the NAD boosting molecule, again has been shown to be beneficial, whereas simvastatin and other statins have not. Lifestyle modification, where only diet restriction has been shown, does not significantly affect the mitochondrial function in long run, especially if the calorie restriction is done, because ultimately the level of free fatty acids goes up. So it has to be coupled with exercise, not only the lifestyle modification. Then there are certain drugs which mimic exercise and they improve the mitochondrial function and fatty oxidant. And these are AICAR, AMPA activators, then PPR agonists, resveratrol, then epicatechin, then imeglimin, and this is a new drug which is tried in India, the TRC150094, and it has cleared phase 1, and it has been shown to improve the insulin sensitivity in rats. Then allosteric activator of SERCA, and finally this is the pictorial representation. There are many class of drugs, mostly experimental, that you can see at that OXPHOS, that is the oxidative phosphorylation modulators, then AMPK activators, PPR agonists, stacks, antioxidants, coenzyme Q analogs, and then novel drugs, MCP inhibitors, MAM modulators. They all can target the mitochondria, they can improve the mitochondrial function, and thereby they can increase the insulin sensitivity and decrease the resistance. So ideal OAD for multifactorial approach in thin fat type 2 diabetes, that is Indians, should target mitochondrial dysfunction, insulin resistance and secretion, and mitochondrial biogenesis, increased insulin sensitivity and increased beta cell function is desired. And final effects can be seen where you can have reduction of blood glucose, blood pressure, better lipid profiles, and better CV profile. So there are unmet needs for thin fat Indians, metformin and then SGLT2. These are the two drugs which have been shown that they can positively improve the mitochondrial function. So mitochondrial modulator sits here in between, and we expect that someday we will have some of the drug which can be combined with these two established drugs to help the patients. So this is my last slide, that with recent advancements in the field of type 2 diabetes, it has become evident that mitochondrial dysfunction is a central element in the disease pathogenesis and progression. Hence, understanding of diabetic pathophysiology at the cellular level, like mine, is a new way forward for novel therapeutic agent development. And with this, we can hope that the aim of therapy of diabetic in coming years shall shift from reducing blood glucose to correction of underlying disease pathology and possibly halting the disease progression. That was all. Thank you very much.